Hello, everyone, and thank you for joining our webinar. Yeah, I appreciate you joining us today. Just a short summary of what we're going to do. I'm David Amaral, and I'm going to start the webinar. I will give a brief overview of the development of the brain, and then we'll talk a little bit about some recent findings that have relied on studying the postmortem human brain to get an understanding of the human brain and of the human brain in individuals with autism. And then I'll pass it off to Dr. Schumann, who will go in more depth about a particular topic of her research, which is looking at the neurobiological basis of anxiety in autism, studying a part of the brain called the amygdala. So let's go ahead and get started. Just a few basic findings or information about the human brain. So the human brain really is a phenomenally complicated organ, part of our body. It has about 100 billion neurons. Neurons are the cells of the brain that do all the information processing. And neurons, you see a picture on the right there, communicate through chemical connections that are called synapses. And you see a picture of that on the left. So there's an electrical impulse that gets passed down the connection from one neuron to the next. And then there's a little gap where chemicals get distributed to the next neuron. And so there's an electrochemical communication. And there's about 100 trillion of these synapses in the human brain. The cerebral cortex, which we think is really important for the symptoms of autism, has about 20 billion of these neurons. And most of these neurons are actually generated during fetal life. And the peak of generation of neurons is between the 13th and 20th week of human pregnancy. And what's astonishing is that during this period, neurons are generated at around 2,500 per second. This is a phenomenon. If you think about something happening 2,500 times a second, it's an incredibly rapid pace. And we're going to come back to that in a little bit to discuss some of the implications for errors in brain development because of that rapid pace. So given the fact that all of this has to happen, the 100 billion neurons have to develop, they have to form connections, they have to form functional circuits. It's not surprising that there are missteps in how the brain develops. To my mind, what is surprising is that it ever develops normally, and generally it does. So a little bit more about brain development. The brain, as I said, all of the neurons of the brain, all of the functional cells of the brain are generated by about the end of the 6th month of pregnancy. There are a few parts of the brain where there's additional neurons generated actually throughout life, but that's actually a rare occurrence. Most of the neurons are generated early on. And at 9 months of age, just at the time of birth, you can see in this picture down below here that the brain has looks like a human brain. It has all the bumps and crannies that we call sulci and gyri. But at birth, the brain's only about 25% of the adult size. And it takes several years to grow to adult size, which generally happens around 6 years of age. You can see that in these images that are on the screen now. These are magnetic resonance images of the human brain of living human individuals, who we can take images of the brain at different times during life. And what you can see, they're all to scale, so that what you can see is that at 1 week, it's much smaller than it is at, say, 2 years or 10 years of life. And what's happening during this early period of development, which is critical for the emergence of autism, which, as many of you know, is generally diagnosed sometime around the 3rd or 4th year of life, is that there's an enormous amount of connections being formed and refined. Some connections are formed, some connections are eliminated. And so it's a very dynamic process that takes place postnatally that leads ultimately to the adult brain. Now, we know that there are many alterations of the normal developmental trajectory of the brain in autism. One of the phenomena that we have been studying here at the Mind Institute for a number of years now is the fact that some children with autism have a brain that's actually unexpectedly large or disproportionately large. So in this image here, these are actually models of children based on MRIs of their brain. And what you can see on the left side here is the size of a typical child. These children are scanned when they're very young, about 2.5 to 3 years of age. And you can see what a typically developing child, what the brain looks like, and you can see that the total size of their brain is nearly 1,000 cubic centimeters. And most children with autism actually have the same size brain. So here's a child about the same age, and their brain size is about the same brain size. But what we found is that about 15% of boys with autism have brains that actually are unexpectedly large. We call it we use the term disproportionate megalencephaly. That complicated term means that their brain is bigger than we would expect for their body size. And what you can see is that this child's brain with autism is about 20% larger than this child's brain and this typically developing child's brain. We're interested in this form of autism because what we know is that these children actually have the worst prognosis. So these are the kids who show the least IQ gains over time. They have the least language function. And at this point, we don't really understand what leads to the enlarged brain. And it turns out that MRI is a wonderful tool for studying some aspects of the development of the brain in autism. But the problem is, and this is now another picture of MRI picture of the brain of a child. Just it's the same brain looking at in different directions. But the point that I wanted to make in this slide is that the smallest pixel, like if you think on a TV screen, the smallest dot on the screen of the TV, you call it a pixel. Well, the smallest dot that in an MRI is called a voxel. It's a volume. And the smallest voxel, or the typical voxel that we have in a picture that we take with an MRI is about a cubic millimeter. So it's this here. And in that 1 cubic millimeter of brain, it would include about 50,000 neurons and 100 to 300 million synapses. And the bottom line is that the resolution of MRI is too low to see neurons and neuron networks. We need to do other techniques in order to understand the brain at a cellular level. So just to show you what the cellular level looks like, these are pictures of the brain of different age children. This was work done many, many years ago by a neurologist named Cornell, who studied postmortem brains from individuals at different ages. And all the black dots here are actually the cells, the neurons, the cell bodies. And what you can see is that at 1 month of age, it's much denser than at 2 years of age. There's more space in between the black dots. And the reason for that is that if we look at a different kind of staining, this is a staining that is named after the person who invented it, whose name is Golgi. These tree-like structures are actually the neurons again. And what you can see is that at 1 month of age, they're very primitive. There's not a lot of branches on the neurons, but over time, the neurons get more mature, they send out more branches, and that's because they're making more and more connections. And that's why the brain is expanding, as I said early on in the webinar, over the first 6 years of life, because the neurons are getting larger, they're getting more connections. And we think that it may be something related to that process of developing connections between neurons that may be altered in many forms of autism. Now, a good example of the benefits of studying the brain come from Alzheimer's disease. Professor Alzheimer, who was a neuropathologist, studied the brain more than 100 years ago of Augusta D., who was an aged woman who had signs of dementia, loss of memory, and other problems. And when he looked at her brain, what he saw was inside of the neurons, there are these processes that he called tangles, neurofibrillary tangles. And he then went on from this first case of Augusta D. to look at other cases and found that older people who were suffering from loss of memory and dementia often had these tangles. And subsequently, people have found that there's another pathological feature, which are called amyloid plaques. And these are accumulations of protein that's not supposed to be there. They cause damage to the brain. They damage connections, they damage neurons. And we now know that these plaques and tangles are the hallmark of Alzheimer's disease. They are actually the signature. And as it turns out, as Alzheimer's disease progresses, you end up having more of these plaques and tangles. And nowadays, people can actually grade how severe your Alzheimer's disease is based on how many plaques and tangles you have. Now, in autism, we don't have this at this point in time. It turns out that the first neuropathological studies of autism weren't carried out until the 1980s. Some seminal work done by Margaret Bauman and Tom Kemper from Harvard University. But subsequently, although there have been many studies that have taken place, we don't yet have a consistent sign of disease like plaques and tangles in the brains of individuals with autism. However, what's interesting is as we are dedicating more effort to studying the human brain, we're starting to have some really interesting insights into brain alterations associated with autism. And I just wanted to highlight a couple of those. So this is actually a paper literally hot off the presses. It really came out online just 3 weeks ago. And this is a paper that was published by one of our colleagues in Autism BrainNet, Dr. Matt Anderson at Beth Israel Hospital in Boston. And interestingly, Dr. Anderson is a board certified neuropathologist. And he basically said, I want to look at brains from the individuals with autism from a classical neuropathological point of view and see if there's anything that I see that hasn't been seen before. And what he found was that there, in 65% of the brains that he looked at, there were indications that there was an inflammatory process taking place where immune cells were attacking some of the, not neurons, but there's a different type of cell in the brain called glial cells. And glial cells in part are protective cells. They actually develop boundaries between the brain and the rest of the body. And what he found was that there was an immune response that seemed to cells, the glial cells, that form boundaries between the blood and the brain and what's called the cerebrospinal fluid. The bottom line is that this is the first paper that has found a consistent neuropathological signature in a very large proportion of the brains analyzed from people with autism. Now, why this is important is because if this can be replicated. And of course, science is always based on replication, but this was a very large study and I think we're optimistic that it could be replicated. This would actually point to the fact that perhaps a portion of autism is due to an immune process. And it would lead us to ask questions about what is leading to that immune process. Is it something that happens early on? Is it an ongoing process? Is it something that could be interfered with? And if it could be interfered with, would that actually help to eliminate some of the symptoms of autism? I think very exciting work, and it couldn't be done unless there were analysis of the postmortem human brain. Another area of research, this is in genetics, and SPARC, of course, is one of the foremost studies looking at the genetics of autism based on taking saliva samples and looking at the DNA in the body. Christopher Walsh, who's at Children's Hospital in Boston, and his colleagues have actually found by looking at the brain that in some cases there are different genetic errors in the brain that are not seen by looking at the blood or other parts of the body. And the reason for this, Dr. Walsh says, is that because neurons are generated so rapidly during early development, sometimes there's errors in the formation of the neurons in the DNA. And it's only errors that are local to particular neurons or a particular part of the brain. And that's called somatic mutations, that they are not throughout the entire body, they're just in one tissue. And Dr. Walsh predicts that at least some individuals with autism, that their symptoms might actually be related not to a general genetic problem throughout the body, but to one of these somatic mutations that's located only in the brain. And again, this would never have been discovered except for studying actual brains from actual individuals with autism. And Dr. Walsh is continuing these studies. So again, we look forward to more information about this. And my final example is not so much about autism, but about the fact that the human brain is, as I said, a phenomenal structure. And while it has great similarities with animal brains, there is increasing evidence that some aspects of its biology are actually different than either mouse brains or monkey brains. And here's one example of it. These investigators who use both anatomical techniques as well as molecular techniques looking at human postmortem brains actually discovered a neuron. These pictures up here are pictures of a neuron. The red one is actually the kind of neuron that they're talking about that has never been found in the mouse brain before. It's entirely unique to the human brain. They call it the rosehip cell because it actually looks like rosehip plants on the beaches in the East Coast. But this has a unique structure and connectivity pattern, and it actually is a cell that is involved in inhibiting activity in the brain, and the investigators think that it might actually have a fundamental role in the regulation of excitatory and inhibitory functions in the brain. So again, the reason that this is important is because we would have never discovered this cell type had we not looked at the human brain. So I hope I've convinced you that by looking at the human brain, we can understand a lot about the neurochemistry and the genetics and the neuropathology of disorders like autism. And really the bottleneck at this point in time is that there just aren't enough postmortem brains to be able to study. We have fantastic techniques. We don't have enough material to actually analyze. The world community of autism researchers really needs more material. And in just a few seconds, I'd like to show you that we're trying to solve that problem by developing Autism BrainNet. Autism BrainNet is is an enterprise of the Simons Foundation Autism Research Initiative. Our goal is to acquire a postmortem brain tissue in order to be able to provide that resource to autism researchers throughout the world. As I mentioned, it's funded by the Simons Foundation. We're a relatively new organization. We were only launched in May of 2014, and we have collection sites around the country. Dr. Schumann heads one here at the Mind Institute. We have 3 other sites around the country. If you want to know more about Autism BrainNet, I would encourage you to go to our website, autismbrainnet.org. You can read about how we function. And I would say that our entire goal is to provide a sensitive and easy way for individuals to consider doing a postmortem brain donation. And we have an outreach manager, Ms. Lillian Acosta Sanchez, who is making relationships with organizations around the country to bring education about the need for postmortem studies. Just to put in contrast, for Alzheimer's, there's been an effort going on for probably the last 50 years. And I would say that tens of thousands of brains have been donated to understand the pathology of Alzheimer's disease. And the reality is that all of the interventions, all of the treatments, all of the preventative measures that are now being studied for Alzheimer's disease are based on studying the postmortem human brain. By contrast, there are only a few hundred brains that have been donated from individuals with autism, and that we continue to need many more donations in order to advance the science. We have a hotline for making a donation, and if you have any questions about Autism BrainNet, you can also go to this. You can contact us through info@autismbrainnet.org. Also, I would encourage you, if you have any interest in this whatsoever, to register. It's really registering for a quarterly newsletter, and you can do that at a website called takebrains.org. It takes 30 seconds to register, and you will then get a quarterly newsletter. And again, there's absolutely no pressure to make a donation. We really want to bring awareness to the autism community about the need and the usefulness of doing postmortem brain research. So now to dig down a little bit deeper into one area where we've made, I think, major advances by doing postmortem autism research, I'm going to turn it over to Dr. Schumann to tell us about the amygdala.
Thank you very much for joining us. I appreciate the opportunity to share our research. I'm going to get into some details of a particular area of the brain that we have learned a lot about over the last few years in studying human brain tissue. But first, I want to give you kind of the take-home point. So mainly, we used to think that very little changed in the brain after birth or after the first few years of life. But what we've learned from our research is that that's really simply not true. The brain is growing and changing throughout lifespan. This is happening in typical development and in people with autism. But what is exciting about this phenomenon is that it opens up opportunities really to discover biological therapeutics that can shape brain development and impact behavior well into adulthood. So this is particularly true for regions of the brain responsible for monitoring behaviors such as anxiety and social cognition, which are common challenges for people with autism and other neurodevelopmental disorders. And these regions such as the amygdala that I'm going to talk about today. But really the take-home message here is that we think that it is the trajectory or timeline of development of these brain structures into adulthood that may be different in people with autism. And if we can understand the cellular and molecular mechanisms underlying this developmental trajectory, and this really opens up tremendous opportunities for us to discover ways that we could perhaps change the course of of development and find treatments for those symptoms. So the example that I'm going to focus on for the rest of our talk is a brain region called the amygdala. It's located straight in from your ear. You can find it there in a region of the temporal lobe. It's the small pink dot there. And this region is involved in detecting potential danger in the environment, orchestrating other brain regions to appropriately respond. You may have heard of fight or flight. So you can think of the amygdala as what we call a danger detector. So it looks for clues for a potential threat in the environment, links this information with previous knowledge, and tells other brain regions how to respond, and then also enhances memory for those highly emotional events. So you can imagine that in humans, this action of searching for clues can become very social, such as looking to the eye region of the face to read maybe what someone is thinking, or finding or indicating a danger in somewhere else. And in fact, the amygdala does play an important role in modulating social behavior. So consider if it's overactive, we may interpret things that are normally benign as scary, leading to feelings of anxiety in everyday situations. But if it's not doing its job, then inappropriate social behavior or even risk-taking can occur. So it turns out that the amygdala takes a very long time to develop. It continues to change into adulthood, and this growth may be influenced by other brain regions and the environment and social interactions. But we think that it may be developing on a different timeline in people with autism, perhaps contributing to symptoms of anxiety. So we first discovered this phenomenon of extended amygdala growth and how that might be different in people with ASD in a series of studies where we took pictures of the brain with MRI and measured the size of the amygdala. As Dr. Emerald mentioned, we can look at how sizes of different brain regions are changing across time with MRI. So what we found is we start to create a map of how the structure is developing. So what we did here is that you could see amygdala size is there on the y-axis and increasing age on the x-axis. And what we found is that in typical development, the amygdala is continuing to grow in volume well into adulthood. This is not something that necessarily happens in other areas of the brain. In fact, we found that the amygdala grows, continues to grow longer than other areas of the brain do. So however, in the amygdala in people with autism, it undergoes a very different growth trajectory. It grows larger early on, but does not undergo the same age-related increase as in typical development. This initial overgrowth may contribute to the anxiety or social impairments in children with autism. So the next question is, we want to know why. Why is it that the amygdala is larger in children? Are there more cells or fibers connecting the cells that haven't been perhaps pruned or shaped properly in the way that they normally would? So then we can ask, why is it that the amygdala is not undergoing the same age-related increase in size as it is in typical development? So it's important to keep in mind that the factors that may make the amygdala larger in people with autism may be different from those that are contributing later in life. So this is why we need to turn to studying brain tissue, because these questions just simply can't be answered at the level of MRI. So we then embarked on a series of studies to find out what cellular and molecular mechanisms make the amygdala larger or smaller. Is it the number of cells or the number of connections that those cells make? So we first asked the question, could it be changes in the number of neurons? As you heard from Dr. Amaral, neurons are the primary cells underlying brain function. This is a cell body there in purple, and the fibers coming off of it are called dendrites, and those dendrites make connections to other neurons. So utilizing brain tissue, we were able to count the number of neuronal cell bodies under a microscope in the amygdala of neurotypical individuals and those who had ASD during life. We did this over a wide age range. So let's start then with typical development. What we found was actually quite surprising to us, in that the number of neurons is actually increasing by about 30% from youth to adulthood in typical development. So as I mentioned, the size of the amygdala is growing, and now we know that the number of neurons are increasing as well. And this, again, doesn't really happen to this extent in most other brain regions. But remarkably, we actually found this to be true in both human and in non-human primates. So we can now add this information to that map of that trajectory that we had showed before, in that seeing that increase in number of neurons in typical development. However, this is not the case in people with autism. So you can see here in this graph, in people with autism, this is mapping the number of neurons in the amygdala, which is there on the y-axis, they're in millions, and the age of the person across the x-axis. And so we had a wide range of ages, going from about 2 years old up to 50 years of age. And we mapped the number of neurons in the amygdala in each of those cases. You can see that in the autism cases are in red, and the typically developing cases are in blue. And in fact, the number of neurons starts out larger early in development in autism, but does not undergo that same age-related increase that you see in typical development. And in fact, we actually see a decrease in the number of neurons. And so the question that we're wanting to know now is why that difference in the developmental trajectory is happening. We published, we can then add this to our map so that if we have too many neurons in the amygdala in children with ASD starting out early and then not undergoing that same age-related increase, and in fact appear to lose neurons into adulthood, perhaps indicating potentially some sort of degenerative loss of cells. So we published this article last year in the Journal of Proceedings of the National Academy of Sciences, and are now researching what is causing the loss of neurons. And of course, we need to have brain tissue to answer those questions. So then we wanted to find out perhaps if it's the connections between neurons that might be increasing and contributing to our changes in volume, and if that could tell us something about the differences in the growth trajectory of the amygdala in people with autism. So there are fibers called dendrites, which you can see there in green sprouting off of the cell body in blue. And we can create three-dimensional maps of the dendritic arbors. So they look like a tree of the individual neuron. We meticulously count those and measure the length of every branch. And what was interesting, and not surprisingly, considering the growth of the amygdala throughout into adulthood and typical development, the dendritic length or arborization of those neuronal connections are growing pretty substantially up into adulthood. We have different ages of 7-year-old, 15-year-old, 31, and up to 44, and there is substantial growth. And so then we wanted to know and look in our cases of autism and perhaps to see if it might be a different developmental trajectory. And in fact, that wasn't the case. In people with autism, those dendrites were growing and continuing to grow at about the same rate into adulthood, despite the fact that there were fewer neurons in adulthood. So we didn't find, or we found that those dendrites were growing at the same rate, but then we wanted to know, what about where the neurons actually connect and send signals to each other? The place where two neurons meet is called a synapse. They are tiny protrusions from this dendritic fiber. You can see there in the picture, the little knobs in the picture, where the neurons actually connect to each other and send signals. So those little spots they're actually called spines. So you can think of it as, if there are too many of those little spots where the neurons are connecting, that you might have too much communication between neurons. And this potentially could, we think, contribute to an overactive amygdala, perhaps too many connections, too much communication may lead to anxiety, or even in extreme cases, potentially seizures, contribute to seizure disorders. However, if there are too few spines, there might be too little communication. And we actually hypothesize, to give the story away a little bit, that perhaps if you have an overactive amygdala, that maybe it could then contribute to some sort of change or loss of spines later on. If you think about an engine running and running and running, and at some point it gives out, that could possibly contribute to the changes across age. So we wanted to ask, do people with autism have a different number of neuronal spines, the place where the neurons transmit signals in the amygdala? So what we found was that when we counted, again, those little individual spines, those knobs on the dendritic fibers, in that you can see on the right, there are two images, one for typical children and one for autistic children, and the colored spots along the fibers are the number of spines, and that there are quite a few more in people with autism, in children with autism, than there is in typically developing children. So we hypothesize again that maybe too much connection between these neurons in children with autism may lead to an overactive amygdala and perhaps contribute to the common symptom of anxiety. However, when we go and look in adults, we actually find that there are fewer spines. So in typically developing individuals, the number of spines doesn't change very much from childhood to adulthood. It changes slightly. But in people with autism, there's too many. And then it appears that there are now too few. So again, we're hypothesizing that perhaps the loss of spines may be due to stress or anxiety or an overactive amygdala. And so the next question is, so we can add this to our map, in that in typical development, you can see the increase in volume, the increase in the number of neurons. But in autism, it just appears to be that there is too much too early. So in a larger volume, an increased number of neurons, an increased number of spines, but then later on into adulthood, it appears that then there's a loss of volume and neuron number and spine number. So the next step is that we want to figure out why it is that this is happening, what other factors may give us clues, and then perhaps what role genes are playing in this growth trajectory. Our goal really here is to find out what is causing particularly the loss of neurons and loss of spines into adulthood. And if we can determine that, then we can start to look for potential treatments or interventions that may change the course of this developmental trajectory. But again, it's important for us to have brain tissue in order to be able to do that. So in summary from this part of our research, so the amygdala undergoes substantial changes in typical development and in people with autism throughout life. It appears to grow too large with too many neurons and spines early in children with autism, which may contribute to anxiety and social impairments, and then potentially loses neurons and spines as people with autism become adults. But the take-home message and the encouraging message here is that there is a lot of change in brain development as typically developing people and people with autism get older. And if we understand how the brain is changing throughout life, then we have the opportunity to change that course and find treatments. So I'll turn it back over to Dr. Emerald to give us some conclusions, and then we'd be happy to take your questions.
Thanks, Cindy. So we're just about done, and we just wanted to have a few general conclusions. The first one is that it's very clear that the symptoms of autism, both the core features and some of the co-occurring conditions like epilepsy and anxiety, are likely due to altered brain development. We don't understand all facets of that, but that's what we're trying to foster with Autism BrainNet and work like Dr. Schumann had talked about. The only way really to understand some of these aspects is of cellular and molecular, at a cellular molecular level, is to study the postmortem human brain. And I think Dr. Schumann gave you good examples of the progress that can be made if you can study the human brain. And again, we want to emphasize that understanding the alterations in brain development and functional neural systems in the autistic brain can lead to more effective targeted treatments for the disabilities associated with autism. Again, just to emphasize that the treatments that are being developed now for disorders like Alzheimer's disease really have stemmed from an analysis of the postmortem human brains. That's also true for Parkinson's disease and multiple sclerosis and a number of other disorders. And we're hopeful that by understanding more about the alterations in the autistic brain, that more effective targeted treatments can be developed. And finally, analyzing the autistic brain provides insight into the way the most sophisticated human behaviors and emotions are produced. So I think it's important to understand that by understanding the autistic brain, we are really understanding the human brain and how it carries out the most phenomenal aspects of human nature, creativity, love, all the other aspects that we think are so dear to humankind are going to be better understood by the kinds of research that we're trying to promote. And with that, I'll say for both Cindy and I, thank you. This is the Mind Institute. This is where we're sitting right now, and we're happy to take any questions from you.
Thank you so much, David and Cindy. Can you hear me okay?
Perfect.
Okay, great. So we did get some questions in. This first one, we had a couple variations on this question, but I think it might be for Cindy. So people were asking about changes in neurons and the amygdala and if that relates to changes in behavior or levels of anxiety.
So I think that's a great question, and actually probably the ultimate question that our research and what we're trying to find out. We are making efforts towards trying to, through Autism BrainNet, through Autism BrainNet, to collect more clinical information as much as we possibly can, and then to be able to relate that information that we can get from those medical records, and then also interviews with the families of the donor, and try to collect that information and then relate it to our findings.
So, and one other thing to add to that is that we do get clues from imaging studies that the amygdala is functioning differently in some individuals with autism, related to to the way faces are interpreted, particularly angry faces or scared faces. And there is a sense that, as Cindy was saying, either the amygdala is too active or in some cases not as active. but the reason for that is still unclear. So I think what we're trying to show is that the reason the people with autism may have anxiety is because there is either a loss of neurons or a altered program of development of neurons. And I think that, that's an important step forward, because by understanding, and anxiety is very rampant. In some of the other studies that we have here going on at the Mind Institute, we found that 68% of people with autism actually have debilitating anxiety symptoms as well. So if we could understand that and even just better treat the anxiety component of autism, we're certainly going to improve the quality of life of many, many people on the autism spectrum.
Okay. Someone had a question about the Autism BrainNet program and what kinds of safeguards are in place for the donation. So keeping them so that, there's not a destruction of the material. So someone I think they're just wanting to understand the logistics and where they're stored and how they're stored.
Yeah, so that's a really good question, and it probably comes from someone who is familiar with the fact that in a predecessor to our program, there was actually an unfortunate loss of tissue, happened many years ago, and we've learned from that. So we ensure that the tissue is stored in highly protected, so some of it is frozen, some of it actually, most of it is actually frozen, and the freezers that we store the tissue in are safeguarded with backup systems. They're locked. There's video cameras on the directed at the freezers as well. So we've made every effort to have the tissue itself be safeguarded, as well as every effort to ensure that the information from the families is safeguarded as well. So when we send tissue to an investigator, for example, we never tell the investigator the name of the donor or the name of the family. It's all coded, and we provide information about whether the individual had autism or not, and whether, for example, they might have had epilepsy or some other condition. But we never divulge family details, so we make sure that that's secure as well. So I would say that we have learned from the situations in the past, and we've made every effort to ensure that the tissue is highly safeguarded. The other thing too is that the tissue is at multiple sites, so we have the safety of having not all the tissue in one place. But bottom line is that at each of the locations where we have tissue, we've implemented very rigorous safety procedures.
Okay, thank you. This could be for David or Cindy. What are some challenges in studying causes of autism? Kind of a big, broad question.
I'll start with one of you think.
Okay.
So, I think one of the challenges is the fact that, some people have said when you've seen one person with autism, you've seen one person with autism. And the fact is that, from Spark and other similar kinds of studies, we know now that there are nearly 200 genes that have been implicated in causing aspects of autism or increasing the risk for autism. Every person with autism, has, of course, the core features of it, but then, 30% of people with autism have seizure disorders. 60% or so have anxiety. May not be the 30% who have epilepsy have also anxiety. A lot of people have gastrointestinal problems. And so there's an incredible sort of diversity, or we call it heterogeneity, about the features of autism. And I think in the end, as I mentioned early on, for example, we found that only 15% of boys with autism have the big brain form of autism. Now, for them, that's an important thing because it means their prognosis is going to be very difficult, and we're trying to figure out how we can help those kids as well. But interestingly, 85% of kids with autism have a normal sized brain. So, that means that when we're trying to study the brains of individuals, we have to get a large enough number so that we can deal with different subgroups of people with autism. And I think that's one of the biggest challenges for doing this, in a genetic study like Spark. The way you deal with that is that you get saliva samples from 50,000 families, so that deals with the heterogeneity. For an imaging study like we're doing here at the Mind Institute, we actually have hundreds of participants that we are able to image. For doing brain research, like Cindy talked about, typically there are far fewer cases that we have access to, and we realize that we're probably missing things because we don't have an adequate number of cases to deal with this diversity or heterogeneity. So I would say the biggest difficulty in trying to understand the causes of autism is the heterogeneity of autism.
I think I can just add to that, or more underscore the point in that because it appears that there is so much heterogeneity in autism, we really need to have all of these different approaches. So we need to be able to answer questions like maybe when and where in the brain that might be developing differently in people with autism. And those aren't the best questions to ask when we're looking at postmortem tissue. That's really important to turn to these larger longitudinal studies like we were talking about with Spark and with the MRI studies. And then for postmortem studies, we need to ask much more specific questions. Questions like, okay, we know it's growing bigger. We know that from MRI studies, for example, and now we really need to know why, and that requires brain tissue to do. So that is one of the biggest challenges, just to underscore what Dr. Amaral said, is that we have to be very careful about the questions that we ask and choosing the best approach in order to answer them.
Okay, thank you. Let's see, what else do we have? So, Cindy, you talked about a lot about the amygdala. Are there other areas of the brain that researchers are focusing on? And if there are, have there been any studies that have used tissue from Autism BrainNet?
So I think there's, yes, there are several studies, and it just happens to be that the amygdala is probably one that we know more about. We've studied it longer than some of the other regions, or more extensive scale in brain tissue. Other areas that have been a focus in looking at the autism brain have been in the frontal cortex, particularly in the outside part of the prefrontal cortex, what we call dorsolateral prefrontal, and then other areas of the temporal lobe, which is where the amygdala is located. And those have been fairly common areas to look at, although we don't know as much about those areas. In some of the cases, we have seen similar patterns to what we see in the amygdala. For example, too many neurons early on, or maybe even a loss of neurons later on in life, but at a different time scale. So it may be shifted earlier and later in different brain regions.
And some people ask, why do you have to do this in the human brain? Can't you do it in a mouse brain? And the areas that have been of most interest to investigators using Autism BrainNet tissue are the areas like the frontal cortex that Cindy mentioned that are really primitive in the mouse brain. One of the most important areas in autism research is a part of the temporal lobe next to the amygdala called the fusiform gyrus. And that's where the human brain does face processing. And as I'm sure many folks know, people with autism tend to not make face contact or eye contact as much as typically developing individuals. And there's the thought that the fusiform gyrus may be functioning differently. There's actually quite a bit of MRI data suggesting the fusiform gyrus is altered. And there are at least preliminary studies, I think more needs to be done, that the connectivity and neuronal architecture of the fusiform gyrus are different in autism. A lot of this needs to be replicated, and we encourage investigators to reach out to us to to see what tissue is available so that we can promote additional replications. Because again, a lot of the initial studies were done on very small numbers of samples. I think Dr. Schumann's studies are an exception that have quite a few cases and controls. In fact, I think her work has the largest number of cases and controls ever done in autism, and we're trying to promote that. We would love other investigators to be able to reach out to us to request tissue and to look at some of these other brain regions in the same comprehensive way that Dr. Schumann has done in the amygdala.
Okay, thank you. We just have time for one last question. What is the criteria to join the Autism BrainNet program?
So a donation can come from anybody who is diagnosed with autism during life. So most of our donations come from next of kin who have a family member who has died, and they either expressed interest themselves or the family has expressed interest in making a donation. Regardless of what co-occurring conditions may have taken place during life, or if it's a genetic form of autism or non-genetic form of autism, any individual who is diagnosed with autism is acceptable. And I should say that we have wonderful clinicians who work with the family in the process of making a donation. There's absolutely no cost associated with it. And the clinicians work through the process of interviewing the family and getting to know something about the donor individual. And we realize that it's a traumatic time in a family's life, and we try and make our approach as sensitive and as caring as possible. And I have to say that we have wonderful clinicians who handle that facet of the process. We are also interested, because we're always comparing individuals with autism to typically developing individuals to see if there are differences. So we are also interested in brain donations from individuals who do not have autism or do not have any other prominent neurological or psychiatric disorder. In this case, we are a little bit more limited in the sense that we are accepting donations from people who are 50 years of age or younger. And we do that because some of the other brain banks in the United States, particularly something called the NIH Neurobiome Brain Bank, has been collecting brains of older individuals for a number of years, and so there is an ample supply of brains from older individuals, but we would accept donations from non-autistic individuals as long as they are younger than 50 years of age. And again, if you go to our website, or if you have a specific question, email us at info@autismbrainnet, then we will be happy to answer any questions that you might have about the process. And thanks again to everybody who's been listening to us. We appreciate you taking the time to join us.
Yes, thank you so much. We are going to wrap things up here. I know that we didn't get through everyone's questions, and I did see a number of questions about treatments and therapies. There are resources on the Spark website, such as webinar recordings and articles about those topics, so feel free to check that